Gervais. I am 36 years old and I have Krabbe. I have been symptomatic on and off since birth. Some of my symptoms include colic, feeding issues, fisting, rigidness, not crawling, and bowel tract problems. I have always had a talent and need for extra sleep. I have a unique relationship with gravity and not picking my feet up well. By the time I was a teenager, my problems were getting worse. I was put on crutches due to excessive falling. At 16 years old, I started right side tremors. 6 months later, I started myoclonic jerks, trouble thinking, and slight vision degradation. Then I was diagnosed with Krabbe and had a cord blood stem cell transplant at Duke shortly before I turned 17. It has been almost 20 years, and I will still come back to Duke yearly for checkups and to help with the unexpected. Without transplant, I would not be here. I would like to thank all my doctors and care team at Duke for saving my life and continuing to provide support when needed.
All right, thank you for coming to session 4, Teaching an Old Dog New Tricks, Cord Blood Banks in Industry. I am very excited to be moderator of the session. When I think about the talks that you're going to be hearing, one of the first talks is truly about how cord blood banks help us. And if you think about it, if the cord blood bank can't give industry or even transplant centers a cord blood unit, then we couldn't save all the lives. So how do they help us do that? So I'm going to go forward here. There we go. So, and I should have introduced myself. Sorry, I'm Heather Stefanski. I'm Vice President of Medical Services at NMDP. Roger Horton is going to be talking about the cord blood bank industry. And so, Roger, I'm going to introduce you. Come on up. Thank you.
All right, let's see if the technology will play ball. Let's press some buttons. Yeah. Bear with. Okay, so I think we've got no conflicts of interest to disclose. So really what I'm trying to talk to you about today is just how, as cord blood banks, maybe can we be a source of expertise for industry partners, because we've got some core skills that I think really are quite transferable. So if we boil it right down, what is it that cord blood banks and their staff do? What is it that our people can already do so well? Well, we work under regulation to produce a high quality product. We are experienced in cell collection and characterization. We've got decades of experience in cell processing and stabilization, and we've got provision of cold chain logistics and product release well and truly sorted out. So who might be interested in those skill sets and why? So depending on how your national regulation works, you may be able to do more with your existing resource to increase patient benefit and also offset your running costs without necessarily having to do extra investment. In the UK, it's possible for us to provide a range of different cellular products under what's known as human application, so stuff for transplant. Cord blood is not classed as a drug in the UK, so it may be a little bit different depending on your regulation area. But we can provide pretty much most things for transplant as long as the process you're doing has been validated and approved by the Human Tissue Authority. We can also provide starting materials for research purposes, process validations and clinical trials, again subject to the correct ethical approvals from different regulatory bodies. So as such, it's possible for us to diversify what we do into sort of multiple products using those core skills without having to invest sort of tens or hundreds of thousands of pounds into sort of new equipment or new licensing and training. So if we just have a look at the journey of the cell in those two different settings. For a transplant, we've obviously got a patient and we've got to have some sort of matching from the register. We get a donor to donate, and then those cells are quality checked and tested. Cryopreservation may or may not come into it, depending on whether someone needs to dissociate the donation event from the clinical transplant. The cells get delivered, and then the patient has a second chance of life. Similarly, for the CGT research and ATMP manufacture, most of those steps are still there. There might just be an extra sort of cell enrichment process or something like that. But by and large, a lot of what you're doing is very similar. One of the first things we kind of got into as a second string to our bow was the cryopreservation step, largely which came about as a result of COVID. because there was a lot of problems with donors getting ill at the last minute and having to drop out. So when the donors were well, the harvest was done, cells were frozen down and then moved on at a later date, just to dissociate and reduce that risk. But cryopreservation of HPCs is not that different from cryopreservation of cord. It's all the same testing, it's the same checks that have all applied before under sort of FACT regulations, FDA, HDA, whatever your local body is. Similarly, if you've got requests for cells for a patient, why can't you have requests for cells from a client? It's the same thing. There are criteria that are required. There may be different nuances depending upon what they're doing with it. But by doing that, you've got the ability to then facilitate research or manufacture an ATMP as well. So if we just have a look at the kind of changing landscape, if we're thinking now how we're going to help out in kind of the adult donor setting, we've got all these different therapies going on here. A lot of them are autologous at the moment, but there are allogeneic therapies coming through as well. But it's quite likely that at some point there's going to be donor cells from somewhere which have got to get to another place for a point of manufacture. If those need stabilization in the middle, we can offer our services to do that. So, and there's loads of these. This is by no means an exhaustive list of what's out there. So if I just give you some examples in terms of numbers and the changing landscape, CAR-T is a good example. We've had steep growth in CAR-Ts over the last few years. So 3 years ago, there was 500 CAR-T products done in Europe. 3 years later, that's 5,000. That's a 10-fold growth in just 3 years. So as more of those ATMPs that we saw a minute ago come through, the numbers are going to go up, and they're going to go up a lot. So this is just an example. This is the last time a new product came on the market. So this is monoclonal antibodies sort of arrived. And for a while it was a bit quiet. But then you look at the growth from, say, 2002 to now. You've gone from a market value of kind of 5 billion to over 150 billion. That's a lot of therapies that are being manufactured. And if a similar sort of thing happens with the ATMPs coming through, we're going to see an enormous growth in collection, apheresis, cell processing, testing. That's all got to happen somewhere. So why, as experienced laboratories, can we not engage in that and help out with that? Similarly, it looks like the US predictions aren't any different. There's going to be quite a lot of products coming through, and it's predicting big growth in terms of the number of ATMPs. So potentially up to sort of 50,000 by sort of 2027 for all indications, and sort of about 20,000 for malignant treatments. So a similar market study done in the US predicted that by 2030, over 350,000 patients will have been treated with a cell and gene therapy product, which, to put that into our sort of transplant perspective, that's the equivalent of all the global transplants that have happened in the last 25 years. And that's what, 7 years from now? We've got to get a lot of extra capacity for stuff from somewhere quick. In the UK, we're expecting to see around about 30 applications for new therapies by the end of the year, and a good chunk of those, nearly 70%, are creating treatments for patients with blood cancer, blood disorders, and related complications around relapse and GVHD. So really what I'm trying to get at is this shift to personalised and precision medicine is an opportunity for us as cord banks to have a greater impact for our patients, but also expand our reach to other patients that we don't currently deal with who can benefit as a result of our expertise. Now, what are the ramifications of this enormous growth? As my boss Dan's keen on quoting, the only thing worse than not having a treatment is a treatment you cannot access. So if we're looking at a 3-4 log growth in the number of treatments that need to be provided, we can't build our way out of that. But we have got a lot of facilities all around the world that are capable of providing something very similar with minor regulatory tweaks in most cases. So we may need to make changes to our approach to licensing, consent potentially to go broader for research and clinical trials. The collection, processing and release testing and sign-off may differ a little bit as well, so we need to take that into account. And we have to be ready to make those changes in order to be able to scale up provision for research purposes, the clinical trials and production of therapeutic products. So for example, this is quite specific to the UK. I don't know whether it applies or not, but we have to add specific categories to our license to be able to do certain things. And if you're going to use advanced therapy, sort of some of your products as starting materials, you have to add that to each cell type. So for example, if you're using lymphocytes for transplant, they're referred to as DLI, but on your license, if you're going to do it for ATMPs, it's classed as peripheral blood mononucleocytes, ATMP. So the usage dictates the tissue type. You'd have to check your local regulator to see if something similar applies, but there's little things like that that have got to be put in place. We're also exploring the possibility of MHRA licensing, which is the equivalent of sort of FDA or sort of TGA in other parts of the world. Because although we can manufacture ATMP starting materials from stuff processed under HTA licensing, it's got potential ramifications on route to market, and it may make extra workload for QPs who have to risk assess it prior to accepting it into the manufacturing chain. If you're all under the same roof, then it makes it simpler. So that's something we're looking at, but it's not something we're doing straight away. FACnet cord, we've already got it, so we're ensuring high quality materials are there. We're also looking at potentially JC accreditation, because the crossover between FACnet cord and FACJC standards, there's quite a bit of crossover there. So again, it makes it easier for us to work with the collection centres that are JC accredited if we're looking at those standards as well. Donor consent needs to be broader, because then we can cover alternative uses in addition to transplant. So if material we collect in the cord bank, for example, isn't suitable for banking, it may well be able to be used in a clinical trial or as a starting material for ATMP. Grassroots research is critical as well. Availability of material is something that academics really struggle with in the UK. And also simple validation of equipment. There's not many places where you can get material to simply validate your cell counter, but you need to. There's also the potential, depending on where you're shipping this stuff in the world, that you might have to consent for additional testing over and above the mandatory that's required by your regulator as well, and that needs to be broached in the consent with the donor too. If you're not comfortable with the idea of broad consent, and not everybody is, there's nothing to stop you going down that route of having a separate donor consent form if that's better. So you can have project-specific consent again if you prefer to go that route. Timings change as well a little bit in the UK. So for transplant, particularly with adult donor material, you can do some of your screening and testing up to 30 days before infusion. But if you're going to use it as a starting material, even though it's collected in the same way, tested in the same way, there has to be day of donation testing. So people need to be aware of that as well, because the idea of urgent medical need has gone. So your timings switch a little bit. Again, that may or may not be something under your national regulator. As we said, the testing, depending on where in the world you are, may change. So, for example, for tissues of placental origin, chlamydia screening is a requirement in Germany. In other countries, you need to do CMV-NAT on the blood products used as a starting material, even though that's not required as the sort of standard mandatory test. Finding CE marked tests for everything can be challenging. So in the UK, we're either CE marked or UKCA. But if it's a pathogen that may not be endemic to the UK, the regulator still wants you to find a CE marked test. So that can be quite challenging, trying to comply with sort of FDA, CLIA, Health Canada, TGA. So you have to try and work your way around that one, or find remote testing labs that can do it for you in the country where you're shipping to. So we are also actively beginning to think about scoping the possibility of a collection site. So we've got 5 cord blood collection sites, but we would actually consider scoping the possibility of an adult donor collection site. Because the issue for us is that the adult donor collections for research or starting materials are currently competing with slots for clinical harvests for transplant. So we need to generate extra capacity there in order to facilitate the research as well as ongoing transplant work. One other thing that we found out, and this was more by way of COVID, was that you need to look at the HPC collection parameters in the same way as we have done for cord blood. So for years we characterize incoming cord blood, we make sure the viability is good, we've got controls over temperature in transit, we know exactly what viability it needs to be and how that's going to be affected. And I don't think it was something that had been looked at that much for HPCs, because only about 1% of products were actually cryopreserved prior to COVID. In the UK before, it was 1% cryopreserved, it went up to 60% overnight. It was a massive increase. So we did a study actually with Sergio Carol and Jesus up at BST, because they got more data than we had. And we found that there were some variables around hematocrit, nucleated cell content, age at cryopreservation, were affecting significantly the CD3 and CD34 populations, but actually differently between the two. Ideally, you need to keep hematocrit under 4%, the nucleated cell content less than 200 times 10 to the 6 per ml at freezing, and ideally freeze between less than 36 hours, otherwise you're going to do more damage. Very quick summary of the results just from the CD34s. So you can see here, by the time you get to sort of 50 hours, you're looking at around about 60 to 70% CD34 viability, whereas if you can freeze it down prior to 10 hours, that viability is kind of nearly in the 90s. Similarly, the CD34 recoveries were dropping off in the same way. CD3 was quite markedly different actually in effects, and actually quite a lot more. So particularly when you consider that sometimes a HPC product may come in, you'll do sort of so many bags of HPC, and then it's not uncommon for transplant centers to say, oh, and whatever's left, can you do a DLI with that? And that usually happens after the first bit. So if you've got a 50-hour product, then you do a 6-hour process, and then you do the DLI, you could be looking at something 60 hours old, which, if you're looking further down the right-hand side of those graphs, that's not a good place to be. You could be looking at viabilities as low as 50% or worse. And in terms of the recoveries post-thaw, not great either. Particularly exacerbated in the CD3s if you're going sort of high-end C per mils, presumably through cell-cell interaction and sort of accidental cell killing from the ligands on the surface. So it was just the same mindset from cord that we carried across to HPC. We just did the same stuff and went, what does it look like? Even CFU, in fact, with the longer transit times dropped off, although CFU being the assay that it is, there's quite a spread there, but there is a general sort of trend down to lower CFU recovery. And again, depending upon what you're treating with a HPC product, that's quite important. Quick plug, if you want to know more, take a picture of that screen. Sorry, couldn't resist. So yeah, those parameters may well be very significant depending upon what you're going to use the product for, as I mentioned. So we need to control them in the same way as we have done for cord blood over all the years. That means that there's the best possible product that we've got for our patients, or also for a customer's downstream application. Some of those other therapies might have additional collection parameters that are going to be required, so we need to be ready to flex for that as well. The processing parameters may be different, so they may want a lower or a higher cell concentration. They may want a different DMSO concentration because that will facilitate a survival of a particular cell population within the product, or maybe some additional additives. In the UK, we would have to do something called a preparation process dossier for that, or a validation for each starting material if it was different than our standard process. But nonetheless, these things are not that difficult to do. They're not hugely time-consuming to carry out, particularly if that means you can then facilitate more products downstream, keep your staff busy, keep material going through the bank. And if you're going to do it across multiple sites as well, centralized and coordinated knowledge transfer is critical, because if you're going to do this regionally, not so much in the UK because we're only a little country, but if you're going to do it across sort of some serious geographic time zones, then you need to have those centralized processes that everyone's singing off the same hymn sheet. Release testing for transplant is very well established. The parameters are well known. We're looking at CD34 and CD45 viabilities, recovery, potency, sterility, that kind of thing. But the advanced therapies may have entirely different endpoints, or you may not even necessarily be able to clarify the endpoints yourself. You would probably have to work with the ATMP manufacturer to say, I've supplied you with this, it looks like this, does it work for your process? And work with them to be able to qualify the starting material itself. Either that, or you may have to develop some different tests that would be applicable to the material. I covered that bit. I hate it when I jump ahead, silly boy. In order to find the relevant endpoints, we need to work with those therapy providers to find out what good looks like, and that's the same picture as we're doing for cord. Logistics and shipment, that's something that we've done for a long time, and that's going to depend very much on the end user's requirements again. It could be 2 to 8 degrees. It could be controlled ambient at 20 to 21, but sort of, you can fix that for up to 72 hours. It could be cryogenic at less than minus 175 degrees. But for most of those, possibly other than the controlled ambient, we've probably got experience before. We've moved DNA samples around for chimerism at 2 to 8. We've certainly moved lots of cord units around at cryogenic temperatures. So we've got the skills for organizing that, and we need to have robust agreements in place with a really good courier that can kind of cover that, or multiple couriers if needs be. That's why we work with specialist logistics providers. It means they can take care of the complex nature of customs. It's also safeguarding the product because it's very fragile, the value of the product as well, particularly to the patient. If it's their one graft, it's really important. So we work with a number of different providers. We might have a different person for hand carriers we use for cryogenic logistics because there's specialists in those areas. They're also better prepared to deal with cryogenic shipments or any problems that may occur in hand carry. You can get real-time tracking, you can have tilt and shock monitoring, and even nitrogen top-ups. If something were to sort of happen where it be tilted in transit, they can give you a top-up. So again, all that knowledge that we've got from moving cords safely around the world, we can apply to those same products. We've got to ensure that the development of any new processes is data-driven, like I showed with the HPC product there. If we control those things, get it right, then the end user is going to get a much better product. They're more likely to come back to use your services again. Streamlining adoption of processes and being aware of requirements is going to help, and it is absolutely critical for the rapid growth needed to promote and provide these materials for research, clinical trials, and ATMP manufacture. So just in conclusion, to round up, since we took on processing of HPC and DLI products, we've released 241. So to put that in perspective, we started in October of 2018 with that particular product. We've currently released 356 cords since 2012. And we've done 241 HPCs in that time. So that's enabled us to help more patients, because there's always a patient at the end of these, because a lot of them have been autologous donations, not necessarily allos as well. And it's also aided sustainability of our cord program and freed up capacity within those partners' facilities as well, so they can look at other things like some of the CAR T cell therapies and develop more stuff. The other thing we found along the way is that a lot of our donors are overwhelmingly in support of actually providing materials for research and clinical trials and advanced therapy manufacture, not just transplant. So let's make it happen and get involved in those therapies.
Thank you so much, Roger. That was a great talk. Now, Dr. Nairi Elwood is going to be talking to us about iPSC cells. Thank you.
Great, and I'd like to start by thanking the organisers and Heather for inviting me to speak in this session today. Induced pluripotent stem cells can be created from any somatic cell by growing the cells in a combination of growth and transcription factors that allows them to be reprogrammed to pluripotent stem cells. These iPSCs can then, under the right conditions, be differentiated into any cell in the body. There are now at least 8 different applications for iPSCs as research products, drug development and discovery, toxicology screening, personalised medicine, disease modelling, stem cell banking, emerging applications, which I find really interesting, looking at clean meat production and wildlife conservation, and what my key interest is in cellular therapy. There are many phase 1 clinical trials using iPSC-derived products and a handful of phase 2 and 3 clinical trials. There are more than 40 commercial companies around the world developing iPSC-derived cell therapeutics, either as off-the-shelf allogeneic or personalised autologous therapies. The diseases being targeted include Parkinson's disease, heart failure, ocular disease, cancer and immune disorders, and in particular NK, CAR-NK, gene-engineered iNKs, graft-versus-host disease, osteoarthritis, critical limb ischemia, hair loss, muscular dystrophy, respiratory distress and failure. And iPSC-derived cells are also being used as a platform technology, such as engineered MSCs as ex vivo gene therapy vehicles. The starting material for creating these iPSC-derived therapies is predominantly skin or blood for autologous therapies, or bone marrow, skin and blood for single allogeneic donors. I think cord blood is actually an absolutely fantastic source of starting material for creating iPSCs. As we know, cord blood is a rich source of immature cells. The cells are naive and have less exposure to environmental and genetic insults. We and others have shown that there's a high efficiency of iPSC generation from just a small volume of cord blood. We've already heard that there's more than 800,000 fully characterised and tissue-type cord blood units stored in public cord blood banks. We don't need to use the whole unit. There's segments available for iPSC generation. All banked cord blood units have a complete family and medical history They've been screened for infectious diseases, have long-term follow-up of the donor, and are of a high quality. The HLA tissue typing of all cord blood units in public banks is known, and so we're able to identify those with homozygous haplotypes, otherwise known as superdonors. And as we've heard nicely from Roger, banks have the capability of storing and shipping iPSCs or their differentiated cells ready for clinical use. So we've been working in this space now for about 8 years, and last year we had 2 key papers published which were really the accumulation of those 7 years of work. The first is the paper on the left, where Pei Tian in my group has developed the methodology, the technical protocol for being able to derive GMP-compliant iPSCs from banked cord blood. And simultaneously with that, Karen Aberton in our group was looking at how to identify which are the good cord blood units to look at and how to go about reconsenting cord blood donors to be able to use their banked cord blood for iPSCs. And I'm not really going to talk about the work that's in those papers, but they're free to download and read. So within Australia, we have 3 public cord blood banks, the one in Sydney, our cord blood bank in Melbourne, the BMDI cord blood bank, and the Queensland cord blood bank. In 2001, these were brought together under the umbrella known as AusCord, and a national cord blood banking network was set up. We work closely with the Australian Bone Marrow Donor Registry, and the bulk of our operating costs are funded by government. All 3 banks are licensed by the Therapeutic Goods Administration, TGA, the equivalent of FDA, and hold FACNet cord accreditation. As of 31st of August this year in Melbourne, we have just over 11,500 cord blood units on the Bone Marrow Donor Registry, and we've released 597 for transplant. So the cord blood banks are very heavily regulated in Australia. As I said, we're licensed under the TGA, and we're licensed as a Class 2 biological for being minimally manipulated, and I can never say that phrase, for homologous use. We have to follow the code of GMP. We have a therapeutic goods order for donor screening requirements, and also a therapeutic goods order specifically for cord blood, which mandates that we have to comply with the FACNet cord standards. And so these FAC standards actually really lend themselves nicely to thinking about banking of iPSCs. And in particular, I think one of my goals is that ultimately our iPSC bank can be accredited under the FACT common standards, because the common standards for cellular therapy really can be applied to any cell source or therapeutic application that are intended to be used throughout product development and clinical trials. So TGA does regulate pluripotent stem cell therapies as a Class 4 biological, as a high-risk product. And that's because we've changed the intrinsic function of the donor biological through genetic modification of the cells, and this includes CAR-Ts or iPSCs. So in thinking about ultimately having this bank licensed by the TGA, we do need to keep in mind that we will have to comply with all the code of GMP and the specific therapeutic goods orders. We also are required to have approval by the Office for Gene Technology Regulator via our Institutional Biosafety Committee, since iPSCs are considered genetically modified organisms. So bringing that back then and keeping all that in mind, we've put a lot of thought into thinking about how we can leverage our own cord blood bank to create cord blood derived iPSC master bank. And so our general sort of working model is that we take one of these representative samples that are stored with the cord blood, expand it up, undergo reprogramming through culturing in cytokines and transcription factors, using the protocol that's been developed by Pei Tian, and we make our iPSCs that we then store as a master cell bank. So in just thinking about the starting material in our own bank, I've already mentioned we're TGR licensed FACT accredited. We have extensive long-term follow-up on all our donors. They've all been screened, they're of a high quality. We know, and it's in one of the papers I mentioned, that we have at least 33 unique homozygous HLA haplotypes stored in the bank. And actually it was interesting hearing from Martin Myers about the homozygous haplotypes in the NMDP inventory, and I did a quick look back at our data, and about 1% of our inventory are homozygous haplotype. We have reconsented cord blood donors to create iPSCs from their banked cord blood, and being part of the AusCord network, we do have the potential to expand this project to include the other two banks. So regulatory considerations, pretty much the same as what we've had to think about for the cord blood bank, and involve looking at our quality systems framework, excuse me, our facility and equipment, and the manufacture of our product. So the quality systems framework for an iPSC bank are pretty much exactly the same as thinking about our cord blood bank, and I won't, for the sake of time, I won't go through all those components, but I do like to put them up to sort of scare people, I guess, who are looking to to move into the GMP space when there has to be a real appreciation of just how much work is involved and what we need to do. And I really like to remind people that it's so important not to underestimate the criticality of the robust quality framework, because that will be key to getting your regulatory approvals down the track. And so looking at our cord blood bank quality system, we've been able to save a lot of money with looking at our cord blood iPSC master cell bank by looking at the documents that we have for our quality management system. For example, we have 121 documents, but 30 of these we've actually been able to pinch and just put a different title on it to use for the master cell bank. And we've only had to create a couple of new documents. It's a work in progress. We're still fully developing the quality system documents, but we're well on the way. Likewise for equipment, there's definitely quite a few documents that we can just use from the cord blood bank. And in production, there's not quite as many, because obviously the processing and storage of cord blood is actually quite different to creating iPSCs. So we've set up a GMP compliant iPSC cell line generation documentation system using lab archives, which our research institute uses, but it's actually been a really nice way of putting all our controlled documents in. We'll still use QPulse, which we use in the bank, but this is sort of a way of having them all together. It also allows for our laboratory administration, keeping track of critical materials and purchasing, and then we can use it as a lab book for the laboratory work as we go through. So Pei in my group has developed a whole lot of forms. You can't do anything without it being recorded on a form that gets put into lab archives. And that's just an example of her cryopreservation record, where every single detail is recorded. So moving on to the facility and equipment, like the cord blood bank, we've had to think about everything to do in the facility and with equipment, from qualification, temp mapping, cleaning, training, security, 24/7 alarm monitoring, our backup systems, and on top of that we've also had the OGTR PC2 laboratory requirements for certifying the lab. So we were able to second a tiny little room within the cord blood bank. So the pink shading on here is the cord blood bank in our floor plan. The yellow is the space that we share with the cell therapy and flow cytometry lab of the hospital. And then the blue is their space. Anyway, we first of all cleared out the old flow cytometer that was in there and have converted that into a PC2 lab, which has now was approved before we started doing anything. And then we've bought all new equipment because we wanted to be able to have a record of every item of equipment right from the day it arrived. And we had to purchase a dual gas incubator to grow the cells at low oxygen. We have an inverted microscope with a camera so we can record. We purchased a new biological safety cabinet that had special glass with holes for the new dissecting microscope to go through, so that this is where Pei and Kawa, who works in the lab, spend many hours picking colonies through the eyepieces there. And this is the testing protocol, the technical protocol that Pei developed for growing GMP-like iPSCs, and it's in the paper, but it's just a summary. We thaw out the cord blood buffy, we expand the cells towards erythroid progenitor cells, then they're reprogrammed, then the virus is removed and placed on vitronectin, and slowly moved to E8 flex medium. The colonies are picked at day 21 post-Sendai reprogramming, and then clones with nice morphology, which are rounded and smooth, are expanded for further testing. And then we verify pluripotency, genomic integrity, and that the cells are virus-free. So that's great, but then we've had to take that purely technical protocol and adapt it to the physical manufacture of, under GMP grade conditions. And so we've done that. All of these aspects, the management of reagents and critical materials, compliance with handling GMOs, traceability, transport, inventory management, end process criteria and criteria acceptance has been done. And then in terms of our release assays, we've relied heavily on the work that we've done as part of GATE, the Global Alliance for Induced Pluripotent Stem Cell Therapies, where the quality control guidelines have been published for clinical grade cells. And so we've set those up. And so for our release testing, the items in red we've set up to do internally. So we look at stemness markers on flow cytometry, and we've been part of GATE with the establishment of a global quality assurance program for these sorts of cells. We check physically that the lines can differentiate towards mesoderm, endoderm and ectoderm, and we also check that the vector has been cleared. And then we outsource to accredited labs our mycoplasma testing, our targeted gene sequencing and SNP analysis, and identity testing. So some of the considerations we've had to think about is, well, what homozygous HLA haplotype will we start with? And this is a table that's in one of those papers. So we know, so this looks at our top 7 HLA homozygous haplotypes in the Melbourne Bank, compared to that published from the UK, and also across all 3 cord blood banks in Australia. And clearly, the winning HLA, is no surprise, is HLA1, B8, DRB13. And then our next one will be 244 and 4. And we actually have figured out through an algorithm that if we made about 5 lines, we would cover nearly 50% of the Victorian population in terms of having a partial match with the population. But then as we're thinking about this long term and applying for grants and so on, I guess we're starting to think about what other ethnicities should we look at. Should we look at Indigenous Australian? We do have a lot of Southeast Asian cord blood units in our banks. Is there another HLA we should be looking at? Where might the global gaps be? Because we do know that other banks, particularly in Barcelona, for example, are doing this sort of work. So, you know, ideally it would be great to be part of the registry and help fill in any gaps. So we're thinking about what will be the best passage number for expansion freezing and to doing all the release test. And I think general consensus seems that it's probably somewhere around passage number 10. And it's important to really define that because you don't want to be doing release testing and certificate of analysis and so on, you know, every passage that you do. How many vials of each line are enough to constitute a master cell bank? Probably 50 to 100, I guess, is what the general consensus is. We do need to think about having backup storage, so you know, storage in 2 places. The release testing, already talked about what we're thinking about in terms of regulatory licensure and accreditation. We need to think up front that we'll need a stability program like we have in cord blood banking. We have a stability program. We've had a lot of thought about the freedom to operate, and that's why we have chosen to use the Thermo Fisher Sendovirus reprogramming kit and other commercial consumables, and happy to chat to anyone about this. I get asked a lot of questions about this, even tomorrow at the poster session. And then business plan, listing on an international register and collaborating with other banks, which GATE is a great way to be able to do that. So we've successfully conducted a full mock run in our new PC2 lab to create GMP mock cord blood derived iPSC lines. We have identified and worked up a cord blood unit that was banked in October 2016. The characteristics of this cord blood unit is that it's female, we've got 5-year follow-up, Northwest European, good TNC, good CD34 numbers. We know it's homozygous by NGS right across the HLA loci, except at DPB1. The next line that we make will be fully homozygous. The cord blood donor's been reconsented. We've sent off a sample of non-viable cord blood buffy for karyotyping and cell identification. There's no evidence of chromosomal aberrations. So we then approved that to get going and make the line. We ordered in all the CTS reagents, and what I mean by that is this cell therapy systems commercially available reagents where they're ancillary materials that have been manufactured with a CGMP for medical devices under 21 CFR, and they've been manufactured at sites that are FDA registered and have drug master files. So although these reagents are super expensive, that's why they're expensive, because they've done all that work, so we don't need to do it. And we're currently at manual passage 3 of 31 iPSC clones following the expansion and reprogramming and picking of the nice colonies. So 21 days after Sendovirus infection transduction, they pick these colonies and manually passage them for 5 passages before then going to an EDTA passage. There have been some challenges, and All the preliminary work that we did with the mock runs used carefully researched commercial reagents that could be swapped out for the fully certified GMP reagents. What we found, now we've actually gone to swap them out, is that some of the companies have slightly changed the formulation of the reagents that we used for the protocol development, the erythroid expansion medium, the vitronectin, from going from the standard or lower grade 1 to CTS grade. They've changed it a little bit. The Sendai virus reprogramming kit, we use the 2.0 kit for our validation and the first runs. They've now upgraded it to a 2.1, where they've swapped out the c-MYC transient gene for a less oncogenic variant of LMYC. And that's all fine, although it appears in our hands that it's decreased the overall reprogramming efficiency when compared to 2.0. It hasn't really mattered. We've still got plenty of clones to work with, but it has been a little bit annoying. And then the short expiry dates of the critical materials and the delays in supplies. It's been difficult to time the ordering of these expensive reagents, and I think you can see it's extremely labour intensive. So the summary, I guess, is what I've already gone through. So hopefully I've been able to show you that public cord blood banks can play a key role in the development of new cellular therapies, right from donor identification, homozygous haplotypes, reconsenting donors, contacting them, making the iPSCs, creating the master bank. That's there then to distribute to clinical programs, industry, to create their end cell of interest. And we have 3 at the moment collaborations in progress where they're working with our GMP mock cells and are just waiting for our fully GMP lines, looking at photoreceptor cell therapy to treat blindness. They have preclinical models all set up to go with these, iPSC-derived CAR-NK and iPSC-derived neural cells. So, I have to finish. It's a very important slide. So there is Pei Tian, who is really the star of all this work. It's her brains and technical perfection that has really allowed all this to progress, and she works with Kawa in the lab, and Karen has helped look after a lot of the ethics and consent work. So I'll finish there.
Thank you so much, Dr. Elwood. That was fascinating. Now Dr. Paul Lin is going to be discussing cord blood-derived NK cells. Dr. Lin.
Okay. Hi, welcome. Thank you so much for inviting me to give this talk. Thank you, Heather, and the organizers of the conference. So today I'm going to be talking about cord blood-derived natural killer cells, specifically for treatment of cancers. So here's my disclosures. So I know that most of you know about CAR T-cell therapy. It has kind of changed the cancer treatment landscape. But right now, the currently FDA-approved CAR T-cell therapies are all autologous, which means that they have a lot of severe limitations. For example, it takes time to manufacture these products, so patients often progress while waiting for their therapy to be manufactured. They've gotten better at manufacturing, but there's still manufacturing failures in order of about 5 to 7%. The CAR Ts, the T cells that are the basis of the therapy are from the patients, and we know that T cells from patients are often defective, so they're not healthy T cells. And of course, because these are personalized medications, they're very costly to manufacture. And CAR T cells have severe side effects and thus are limited to specific institutions. However, T cells are not the only immune cells in the body. If we go back to the diagram of hematopoietic stem cell and differentiation table, you can see that T cells just make up one of our many immune cells, and there are many other immune cells that we could possibly work with. And in fact, people have. And so here's a list of some of the different types of immune cells that people are looking at to kind of use for cancer treatment. And they are at different stages of clinical development. And each of them have their own unique features, kind of what makes them special, about whether both on the positive aspect as well as maybe why they're not as good. The furthest one along right now would be natural killer cells or NK cells, which will be kind of the focus of my talk. So kind of give a little bit history about what natural killer cells are. So unlike T cells, which get activated through the T cell receptor, natural killer cells are activated by kind of a balance of inhibiting and activating factors. So if you have an overwhelming majority of inhibiting factors, then the cells will not be activated. In contrast, if there's a lot of activating factors, it can tip the scale over and activate the cells. Here's a list of just some of the different activating, which are up here, inhibiting receptors that we know exist on NK cells. Some of them are very familiar to us. Some of the inhibiting factors, for example, TIGIT, TIM3, LAG3, those are also checkpoint inhibitors, inhibiting factors for T cells that are also present on NK cells. But you'll notice that there are other ones, a lot of them involve HLA molecules, which will come important in just a little second, that are severe inhibiting factors to NK cell activation. And NK cells also have CD16 receptor, which also come in just a second. So what are different ways of activating NK cells? So under normal conditions, if you have a healthy cell and the NK cells kind of interact with a healthy cell, you'll have an activating, there'll be some kind of stimulatory ligand, so that will be an activating factor. But you also have an inhibiting factor, and one of the strongest inhibiting factors for NK cells is the MHC molecule. So healthy cells will often have MHC molecule, which would be a strong inhibiting factor that will prevent the cells from activating. Now in part B, one of the major things that cancer cells use to evade T cells is to lose their MHC molecule, right? But I think of NK cells as acting kind of as a check to the T cells, kind of working in partnership with T cells, right? So TCRs require MHC molecule to become activated. Tumor cells can try to downregulate MHC molecules to prevent this T cell activation. However, that eliminates one of the strongest inhibitory factors of NK cells, and thus, in the missing self-activation theory, NK cells become activated and will kill the tumor cells. Now, of course, if you also have stressed or damaged cells, that will upregulate a lot of the stimulatory factors that will overwhelmingly tip the NK cells to activation and tip the cells and activate them as well. And then finally, there's something called ADCC, which is antibody-directed cellular cytotoxicity. In other words, because of the CD16 receptor I mentioned, it can bind to the Fc portion of antibodies, and thus any foreign molecule, or in this case, tumor cells that might have an antibody that attaches to it, it will become activated through this receptor. There are other ways of enhancing NK cell activation as well that we can artificially induce. One is using an antibody to kind of block inhibitory receptors. Similar to T cells, we can use, like, for example, PD-1 inhibitors to kind of block this inhibitory signal to, again, tip the NK cells towards activation. We can try to use an allogeneic NK cell, in this case, so that there will be the NK cells will recognize the foreign tumor cells as a non-self, and thus it won't have a strong inhibitory factor. Another way is if you have some kind of bispecific binder, just like you have bispecifics with T cells, you can have the same thing with NK cells, and that can also activate NK cells. And finally, again, similar to T cells, we can put an artificial receptor on the NK cells, a chimeric antigen receptor, a CAR, and that can also activate NK cells. And so why are NK cells better than T cells for CAR therapy? Because NK cells can be allogeneic product, it can be off the shelf, decreasing manufacturing time, decreasing the time the patient has to wait to get the therapy. Because again, it's allogeneic, we can create multiple different doses from just one patient, thus potentially significantly decreasing the costs. From studies so far, we have not found allogeneic NK cells to elicit a GVHD reaction, like T cells. And because of the low toxicity, it means the NK cells can be more widely available. You'll have to go to specialized centers. Potentially, you can even think of using these cell therapies as outpatient. So you can think of like a community doctor can potentially deliver this therapy. And NK cells, kind of we mentioned before, if you put a CAR, for example, in NK cells, the CAR will, of course, activate the NK cells, but you also have the NK cells' natural ability to become activated against tumor as well. So you kind of have two mechanisms of activation for the NK cells. Now, we can get NK cells from a variety of different sources. You can collect them from the peripheral blood of adult patients, for example. We can have a cell line that we can develop. We can collect them from umbilical cord blood. We can generate iPSC cells, kind of what we just mentioned over here, and differentiate them into NK cells and use those as well. And each of them has their own pluses and minuses that I won't really get into. But for the rest of the talk, I'll focus on umbilical cord NK cells, which is what our group at MD Anderson, what we mostly focus on and work on. So MD Anderson has a cord blood bank established and led by Dr. E.J. Spall since 2005. We've collected thousands of different units. We collect multiple units every day, so we have easy access to these. And of course, you can generate multiple doses of these CAR NK cells from just one cord blood unit. And so at our institution, we conducted one of the first in human studies using CAR NK cells in patients to treat B lymphoid malignancies. Leland had kind of talked about this, introduced us to this earlier today, so I'll kind of go into this in a little bit more detail. And so in this clinical trial, we had cord blood-derived NK cells. We would transduce them with a retrovirus to generate a chimeric antigen receptor on the NK cells. We would expand these CAR NK cells, and then for the patients will be conditioned similar to CAR T cell patients with flu-cy. And then we'll give the patients one infusion and multiple different dose levels as part of the clinical trial. The construct is shown here of the CAR that we used. It has ICAS9 suicide switch, which means we can kill the cells if needed, if there's severe toxicity. We have our binding domain, which is a scFv to CD19. And then we have our two co-stimulatory domains, CD28, CD3 zeta. And then unlike T cells, for NK cells, we have to put in an IL-15 cytokine to kind of enhance their function, increase their persistence in the body. And so this was recently published, it's been a few years now in the NEJM. And here are the patients that were treated, that were listed in the publication. You can see that these patients had multiple lines of therapy, 3, 7, 4, et cetera. A lot of them had transplant already, for example, and they still progressed, right? And what we found was, in terms of toxicity, you can see that none of the patients had any CRS or neurotoxicity in contrast to CAR T cells. And then toward later patients, so these initial patients were HLA matched. The last two patients were not HLA matched, and we still did not see any CRS or neurotoxicity. And we had, in fact, great response. A lot of patients had CRs, leading to an overall response rate of 73%. This is an example of one of the patients pre-admission. You can see that disease is everywhere, and then 30 days post-admission, the disease had resolved. And these CAR NK cells were detectable in the blood for up to 12 months post-infusion. And you can see in terms of adverse events, there were very little grade 3 and grade 4 adverse events. Most of the grade 3 and grade 4 were hematologic, which is to be expected from our conditioning regimen. But otherwise, it was very well tolerated. So one of the things we learned from this treatment was that all cord bloods are not the same. And so this is kind of the initial studies I'm showing here. We found that there are some cord bloods that perform better than other cord bloods. And so here's an example in the mouse study where we tested this in the Raji lymphoma model, where here you have tumor alone. You can see that the mice all die by around day 21. And with a suboptimal cord, the mice do survive longer, but they still die shortly after. In contrast to if you find a perfect cord, then the mice live a lot longer. And if you do genetic analysis, you can see that there's a difference in the transcription factors, for example, between the suboptimal cords versus the optimal cords. Right? A lot of the FOS genes, for example, is upregulated in suboptimal cords in contrast to cord bloods. So we're kind of looking at that to make sure we pick the optimal cord bloods for use in our treatment. So I'm going to kind of give 3 additional examples of NK cell therapies that our group at MD Anderson that we've worked and developed amongst the multiple different ones that we have ongoing. So one of the projects is developing a CD5 CAR NK cells against T-cell malignancies. So you can think about it's kind of difficult. It would be kind of difficult to develop an anti-T-cell if you do a CAR T-cell against a T-cell malignancy, right? Because if you develop a CAR against a T-cell, then the T-cells will kind of kill itself, right? So that's one advantage that NK cells have. So here, CD5 is a marker that's present on T-cells, and so we can put this on, develop a CAR against that for NK cells and thus not have fratricide. And so that's what we did here. This is some of the preclinical mouse work. When we have a CAR NK cell, we have prolonged survival that led to increased survival against T-cell cancer. And now we are working on putting this into a clinical trial. Now, can we further enhance these CAR T-cells by targeting checkpoint inhibitors? So checkpoint inhibitors, the traditional checkpoint inhibitors that we think about, PD-1, TIGIT, LAG-3, for example, but we can be a little bit more creative than that. So one of the checkpoint inhibitors we looked at is something called CISH, or cytokine-induced SH2 containing molecule. So if you can remember back earlier in my talk, I talked about how these NK cells have IL-15 to kind of enhance their function. Well, IL-15, when IL-15 is present, it kind of induces a negative feedback loop through this molecule called CISH, right? So this molecule is rapidly induced in response to IL-15, and it gets activated by IL-15 to downregulate NK cell activation. So our thought was, well, what if we knock this checkpoint inhibitor out? Will the cells become more functional from the IL-15? And this was published in Blood by one of our researchers in our department, Dr. Dyer. I'm going to skip this part and go into the mouse studies. And so you can see here, again, in the Raji model, here we have the CD19 CAR by itself, and it does prolong survival in the mice. However, if we also knock out the checkpoint inhibitor, we were able to significantly increase survival. And here's the Kaplan-Meier curve. You can see in the purple that if we knock out the CISH, we can significantly improve the survival beyond just the CAR itself. And the final example of NK cells I'm going to talk about is using bispecifics with NK cells. So one of the bispecifics we looked at was a bispecific called AFM13. So AFM13 is a bispecific targeting CD30 on one end and has targeting CD16 on the other end. And as I mentioned before, CD16 is present on NK cells. So this acts kind of like a bridge between the cancer cells and the NK cells. So this molecule, before we looked at it, was already in clinical trials and was not actually showing very strong efficacy. So when the company tried this in patients, a single agent, AFM13 alone, they were getting response rates in the order of around 10%, right? So not great. But our thought was, well, what if you use allogeneic NK cells? And if you remember correctly, when I mentioned before, with like autologous CAR Ts, these T cells and immune cells from a cancer patient are very dysfunctional, right? They don't work properly. They don't function well. And so if we give healthy NK cells or healthy immune system cells, then they might work better and kill better. And so we kind of see this. So we were able to, and then another thing we did was we kind of pre-complexed these NK cells with this AFM13 prior to infusion to kind of make it kind of CAR-like. So coat the surface with AFM13 prior to infusing. And then we give additional infusions of AFM13. And when we did that in the mouse studies, we saw increased survival of AFM13-loaded cord blood NK cells. And so we started a clinical trial. This was led by Dr. Iago Nieto, in which we have cord blood NK cells. Another wrinkle is we also pre-activated them to create kind of like a memory phenotype with a certain mix of cytokines. We do the conditioning regimen again. We infuse AFM13 coupled with NK cells to create kind of like a CAR-like NK cells. And then we infuse AFM13, continuously infuse AFM13 on a weekly basis. And so these patients, we've treated 41 patients. You can see that these patients were, again, very heavily pretreated, average median of 7 lines of prior treatment. All the patients have had some kind of anti-CD30 antibody treatment with brentuximab. A lot of them had also prior anti-PD-1 treatment as well, and a few of them even had CAR T treatment. And all of them, had progressive disease, except for one patient who had standard disease. And we saw a significant response. So of the 40-some patients, here reporting 40 patients, 37 patients had some kind of response, leading to overall response rate of almost 93% with 65% CR. And then when during the dose expansion phase, with 34 patients, you can see that the overall response rate increased to about 94% with a 70% CR. Again, in the situation with allogeneic NK cells, we saw no cases of CRS, neurotoxicity, graft versus host disease. Here's the waterfall plot for treatments, and here's an example of one of the patients that had numerous lesions prior to treatment and 30 days post-treatment. Had CR. And this patient had multiple lines of treatments, you can see here, including allogeneic stem cell transplant treatment and CD30 CAR T cell treatment. So in summary, we can place chimeric antigen receptors on different immune cells, such as natural killer cells. Clinical trials are ongoing with allogeneic NK cells, and they show promising results even in patients who have been heavily pretreated with very minimal side effects. We can further enhance these CAR NK cells by using CRISPR gene editing, for example, modifying and editing out checkpoint inhibitors. We can do this across multiple different antigens. We have multiple in development. I just mentioned a couple in this presentation. And of course, we can combine NK cells with bispecific engagers to enhance their tumor potential. So unfortunately, my acknowledgement slide didn't make it to this presentation. But of course, all this work was done by a number of different people. Our research group has grown to now over like 60 people in our group, and all of them contribute a lot. Special shout out to Dr. Katie Rizvani and Dr. May Dar, who are kind of been my mentors through this whole process. All right.
Thank you so much, Dr. Lin, for that excellent talk. This session is now open for questions. I have a question. I'm going to start off with a question. This is for you, Dr. Elwood. So I'm curious on how did you, or maybe should I ask this differently? Did you have to convince the cord blood bank to take on iPSC cells? That seems like a big undertaking with just the amount of work that would be, and then even finding space for it in the hospital, like a lot of negotiation.
So I also run a cord blood stem cell research laboratory, and so that's really where this started. All that GMP-like work was done in the research lab with just small philanthropic grants that we got, and it helps when you're the director of the bank because finding the room to then move to the full GMP, that was a little bit of a challenge because I couldn't manage to coerce one. Well, even in the research institute, it wouldn't have been right. So we just had that space that the old flow cytometer was in, and so we just took it. And no one's really said anything, I suppose. And you know, we've had good buy-in from the cord blood bank staff, who are all the other people in that photo that I showed. And we employ, so Karen Abberton works 1 day a week in the research lab and 2 days a week in the bank. And so there's a bit of crossover with the consent and the ethics and so on. And so that's how we've managed that, yeah.
Awesome, thank you. Go ahead. Question, Joanne?
Hi, this is for Nairi. Thanks for your talk. I wondered if you could talk a little bit more about how you genetically screen your iPSC before you plan release, because I thought I saw WGS on your slide, but
it should actually say targeted genome sequencing. It's not whole genome sequencing. I think the correct term is targeted, because on the initial sample we've just done very high level, you know, just looking for chromosomal rearrangements, and we didn't expect to find anything because there was nothing in the family history. But the whole purpose of doing that is so that when we have our master cell line, we can then check that it hasn't been screwed up by the process. And so what we were planning to do there is we had initially planned to just look at about 20 genes that might be perturbed, and you know, your classic sort of oncogene ones, but we will be using one of the commercially available arrays. So we'll be probably, you know, I think 800 or 1,000 of your sort of common ones that might be upregulated or downregulated. So, yeah.
I have a comment to it, because do you do also chromosomal analysis on your starting material, that means cord blood? And in how many cases do you find the aberrations?
So in the you know, all the pre-work that we've done, we do find chromosomal abnormalities after, not in the starting material. So in the starting material, we haven't looked at that many. We've really just targeted the cord blood units, you know, the 5 units that we've looked at so far.
Oh, okay.
But after reprogramming, we do occasionally see chromosomal abnormalities, and so obviously we don't continue with those. Yeah.
So working in Europe under ATMP guidelines since many years, we are required and have to exclude all cord bloods with a small translocation, and you would be surprised in how many cord bloods you see a small chromosomal translocation.
Yeah.
You see them also after reprogramming, but this is normal. But before reprogramming, for me, it's a question. We have to discard all these units in a full ATMP compliant product, because we cannot interpret whether this chromosomal translocation, which is sometimes minor, has an effect on this product. So we are obliged, and therefore a mosaic chromosomal screening is extremely important before you go more or less into reprogram, because it saves a lot of money.
Yeah. Yeah,
and
and a few years ago, we actually did do a study where we had a multiplex PCR set up to look at TAL-AML and a handful of other chromosomal rearrangements, and we did actually find that 1 in 100, so we did it over a whole year, and we did actually find, you know, the TAL-AML rearrangement there.
Yeah, but I don't talk about 84-6 strands primary translocation. I'm talking about other minor translocations which occur in other chromosomes.
Right.
And so it's just extremely relevant to screen them out, because it costs a lot of money.
Yeah, and that's why we're doing the pre-donor testing at a higher level, but the whole purpose is to then just know, at least at that higher level, that they're okay. But I do understand what you're saying. You might miss some if you're not doing, you know, targeted genome sequencing right the way through. But, you know, I guess that's the beauty of having all the family history and so on, which also helps if there's no sort of medical history of anything. So.
Just a comment, you don't see it because it's spontaneous during fetal development, and it occurs because even in the fetus you get a lot of translocation, mainly based on the birth. And there are very good studies out showing that cord blood has a low mutation rate, for instance, and translocation rate as compared to other sources, but still there are a lot. And the question for us will be in the future how we deal with this interpretation of the small translocations. What does it mean? Is it relevant? Is it relevant for the iPSC? Yep. And under the regulation we have in Germany and in Europe, the regulators say you cannot release this cell line. Yeah.
Yep. Thanks.
Hi, thank you, Nairi. Sorry, another question for you. And forgive me if this is a naive question. Do you anticipate that your individual iPS clones are going to have been derived from different types of cells in the Buffy coat? And could that have implications for the different clones activity or use?
But by the time we get to the reprogramming step, it's a pretty homogeneous population of erythroid progenitor cells. So when we started out on this, we thought we would derive 34s and transduce those. But actually at the Institute in our iPSC core facility, their protocol is to get erythroid progenitors, which is what we started to do. And it works so well that we've just stuck with that. So there probably are sort of subtle differences, but we check by flow cytometry that the bulk of the population fit within a certain erythroid progenitor population.
So, hi, I have a question for all of you and then one specifically more for Paul at the end. They were great talks, thank you so much. I'm curious what you guys are thinking about in terms of, you know, vision, especially for the iPSCs. And, you know, you're creating these lines and where do we see them being used? Nairi, you said something, I can't remember, was it 5 lines for 50% of the population? I mean, some, and I assume you were talking about thinking of stem cell transplant for those or for matched thing?
Sorry, just for being, so the end, the iPSC derived cell from that, you know, from 5 to 10 lines, we know that it would match almost 50% of the...
So for any cell therapy?
Yes.
And I guess the reason why I'm bringing this up is it's at least it seems like in the United States, the last few conferences I've been to, iPSCs are, you know, they were the sexy cell, and now they seem to be falling in their popularity. And I'm not an iPSC expert, so I don't understand why. But when you think about, you know, this was brought up in the first talk, these therapies have to be available to patients.
Yeah.
And when we derive clonal cell therapies, in the allogeneic setting, my concern is that you can only give that to a patient 2 or 3 times before they become, allosensitized to that. And so I'm just curious what your thoughts are around how do we, like, it's great, right? It was the cost of goods. How do we make these things? How do we get them into patients? And iPSCs were the thing because they were a factory. They could make you as many cells as you wanted, but that is not going to overcome the barrier of allosensitization if they're clonal.
Yeah, and I guess I haven't really thought about that, but I suppose I would be thinking that this would be a one-off therapy, for a patient. So I'd have to think about that more.
Yeah, because I think, Roger, this is like our, right, we need to get these cells out there, and it's an allogeneic therapy, right? So I think that's our biggest problem is lack of in vivo persistence. And so, we've been focusing more on how do you derive a cell type from multiple donors so that you always have something to give to patients, and we have to be able to cryopreserve them, right? So that was the other thing, and that was my question for you, Paul. If you didn't mention anything about cryopreservation, and if we're going to deliver allo cell therapies, we must be able to cryopreserve them.
Yes, you are absolutely correct. And so when the CD19 trial that I presented was first published, at that time, this was a while ago, I think the trial started in 2017, 2018. We had not developed a way to freeze the cells, and so all the cells were given fresh. And so, I think there's a couple ways to think about that. One, you can think of it as, I think someone mentioned it, like a baker, right? They're constantly baking new cells, and so you can give them, they'll be available every day, and so then you can ship those off and use them right away. That's one way of doing it. But on the flip side, we've done a lot of work, and now we've found a way of freezing these NK cells. And so a lot of our trials going on right now are actually using frozen product. So we've gone through multiple iterations, and we found a method that seems to work. And so you're right. So now we do bank them. Like the most recent trial, the CD70 CAR NK cells, we used 2 different cord bloods, and we banked all the doses we need for the whole clinical trial. And so we've already got all the doses ready.
Too, right? These aren't, unfortunately, they're not T cells that love to, expand, massively peripherally, right? These are NK cells, so they won't expand in the same way, right? Because so you'll have, I assume you'll, although I guess CAR maybe you won't have to do multi-dosing, but it's probably not one and done.
Yes, so the studies are one and done, exactly. Versus, for example, like the AFM13, we subsequently did have to do multiple doses because they don't have anything. Like I said, mentioned in the CAR19 trial, we did see the persistence up to a year, and we still saw the cells in the patient's body. So the theory is once we have the CAR, especially with the IL-15, which is a key, that drives persistence. And we recently published an article. Well, there are other thoughts that we're working on about why they are persisting in addition to the IL-15 that hopefully we'll be able to present later on. But great question.
Thank you, Paul. It was a great talk. And thank you also, Paul, for showing all the receptors on the NK cells. And there are different receptors out also in T cells. And when I see the presentation, and this is also coming back to the clonal ones, first of all, if you reprogram an iPSC, and the iPSC is remembering the background she is coming from. So if you want to reprogram and get an NK iPSC, you also should think about the receptors, because the receptors are the thing who are doing the job in the patient then laterly. And this comes back to the question of Colleen. And the same is true because there are also people out who try to get T cells, but also the iPSC derived should come from a gamma delta T cell or T cell, because this is immunology. It reminds the background is kept more or less. You cannot change it. So if there would be a source which would be good, you should look for a cell which can do more things. So then if you go to an iPSC and not restrict yourself, because in immunology, all immune cells are coming from a stem cell, and you need a background of a cell, and in your case, NK cells. So it's fascinating. And I have another question. What about the studies who are going on the CD16 receptors? Because there are also, maybe you can comment on it, there are also people out to try now to combine CD16 to enhance the cytotoxicity of the NK cells, I think.
So you're talking, just to clarify, CD16, right?
Yeah, right.
Yeah. So that's the receptor that binds to antibodies.
Yeah, right.
There are different variations of that. Certain ones bind stronger than others. So I know like Dan Kaufman has worked a lot on trying to enhance that and create better ones. So that's definitely, yeah, so that's one possibility. You could definitely modify the receptor to have enhanced NK cells, again, with combining them with antibody treatments, yes.
Is it possible with cord blood too?
Sorry?
Do you work with cord blood too then? Do you do the studies? No.
Cord blood, sorry, so you mean put the CD16s on cord blood?
Right.
So you can, yes, you can screen cord bloods for high affinity CD16 variations as well, yeah.
Okay.
So yes, you can do that, just exactly.
I have a follow-up question for Dr. Lin. So I was also thinking about this with the NK cells. You said that some of the batches were more effective than others, and I was wondering if you look at the affinities for class 1 in the host, or in the recipient, and does that correlate with their efficacy and their ability to become licensed?
So it's kind of a new line of work we're working on. We're looking at a number of different factors to try to determine it. We're taking these different cores. We're looking at the different surface markers. We are, doing RNA-seq and looking at the ATAC and all this other stuff. It's still in the very preliminary stages, so I don't have anything to comment at the moment. But all the things you're talking about are stuff we're looking at.
You didn't just simply look at the affinity for class 1?
We're looking at everything, yeah.
Okay, thanks.
Thank you so much for coming, and I believe it is the reception next. So thank you.